This has been a very exciting meeting at ASH in terms of preclinical studies in multiple myeloma. There's been a lot of new energy and a lot of new data presented in terms of the molecular characterization of multiple myeloma at the level of next generation sequencing, including single-cell sequencing studies that provide information not only on the molecular features of the tumor cells, but also on the molecular features of different immune populations. And this topic is obviously very relevant given the progress that has been achieved in the last few years with the development of several new forms of immune-based approaches for the treatment of myeloma, but also other hematologic malignancies. Another very important area of development is at the preclinical level, the new studies in terms of new agents that intend to enhance immune responses or achieve immune response in the form of immune effector cells, such as different forms of chimeric antigen receptor T cells or chimeric antigen receptor expressing natural killer cells, and also bispecific antibodies. And these two areas have had obviously important progress in the clinical arena with clinical presentations about those classes of agents, but also development of new ones that were described in the preclinical sessions. From the preclinical standpoint, there have been also new studies in terms of the development of novel genetically engineered mouse models and the use of these models to understand the biology of the disease. And an important example is the work presented by the group of Jonathan Licht from the University of Florida, describing the development of a new model of myeloma that's based on the LGP130 system, with superimposed loss of function of KDM6A, an important tumor suppressor for this disease. This is an important model because it is expected to provide opportunities for additional functional studies that will interrogate the disease in a more systematic fashion. Finally, and not to tout my own horn, my own laboratory has been conducting extensive studies that we're presenting this meeting on the use of CRISPR-based functional genomic technologies to interrogate the function of genes that drive myeloma or suppress it. We've presented work on the use of gene editing technologies and reported that a very large number, in fact, the majority of genes that are recurrently mutated in myeloma, when we examine them in cell lines in the laboratory, it looks like most of these genes don't increase or decrease tumor growth, so their potential role in the myelomagenesis process may involve other events, probably related to the interactions between the tumor cells and their microenvironment, DNA repair, or other processes not directly related to proliferation and survival. And also we present data about the use of new CRISPR-based approaches, including CRISPR activation to induce genes at will and see what the role is in myeloma, and the use of new dual knockout approaches with CRISPR to see how different genes interact with each other, and how these systems can be applied to better understand mechanisms of resistance to novel class of therapeutics such as the pharmacological degraders of key oncoproteins and natural killer cells. So it's been a very exciting meeting with a lot of new directions, both from the therapeutic side, development of new models for preclinical evaluation, and also new molecular profiling approaches that have been applied to better understand the disease.